from, back then from Technical University of Denmark, but now he works as a development scientist at Cell Bank at Novo Nordisk. I will introduce Morten a little bit. So he has a master's degree in molecular medicine, actually from Aarhus University, where he has done research projects in collaboration with iNano and molecular biology during his time at Aarhus University. Afterwards, he joined the Technical University of Denmark for his PhD, is where we actually met at the Department of Health Technology. And like I said, now Morten works at Novo Nordisk, but the talk today will be about his PhD project and the recent publication on development of cell-based tissue models for testing oral drug delivery devices. I personally look forward to hear about this. So please, Morten, continue.
I will try to share my screen. This, can you see the screen? Great. Yes. Okay, so this talk I've called Connected In Vitro Tissue Models for Oral Drug Delivery. And it covers the work that I did while I was employed at TCU as a PhD student. And the outline of this talk is that I will start by some introduction, and then I will go into the actual results of my PhD research project and cover these 5 areas, which is biocompatibility and stiffness of tissue models and the specific tissue models and coupling of the tissue models and mechanical actuation of the tissue models. And this part about stiffness and mechanical actuation, I will touch very briefly upon and focus more on these other 3 areas. And then I will round off with a conclusion in the end. So the focal topic of the group that I was employed at DTU was drug administration, and specifically oral drug administration, which is a very large research area and also area of focus in the pharmaceutical industry, as 60% of drugs are in this category, and also this drug route has some benefits in terms of patient compliance, and it is also easier to take a tablet than to have an injection. So if you look at the pharmaceutical industry, then it's very difficult to launch pharmaceuticals. And that is because you start out with a lot of drug candidates, and then you go through whole like roll of studies. You go through in vitro studies, in vivo studies, clinical studies, until you finally can launch one compound. And this whole journey is very expensive. So if we look at my PhD, then I was working in this area of in vitro studies, trying to improve the in vitro studies so that you could easier select better compounds. So instead of maybe selecting 250 compounds, you theoretically should be able to narrow it down to fewer, maybe 100, and then it will go faster in the in vivo studies and the clinical studies, and be more, be cheaper since you are testing out fewer compounds. That was the idea of my PhD project. And then if you look at the position that I have now at Novo Nordisk, then I'm more in this area where we are responsible for some parts of the starting materials for the drug substances. Yes. So how does an oral drug reach systemic circulation? That is kind of the starting question of my PhD, because before that I could start to sort of simulate how or simulate absorption of an oral drug, I needed to look into how an oral drug would actually get into the body, so I knew what I was supposed to simulate. And the route from the oral route to systemic circulation is, of course, through the mouth, and then the drug travels to the small intestine, where it is absorbed into the body. And once it reaches the small intestines, the drug can then travel into the bloodstream, and via the bloodstream and the blood vessels, it will travel to the liver, where it will undergo first pass metabolism. So the interesting, or the thing that you want to know when you're developing an oral drug is how much is absorbed and how well is it distributed and how much of it is metabolized in the liver before it is able to reach circulation and have an effect on the body. So if we start by looking at the small intestine, what then is important of the small intestine is that it has a large surface area, and then it is that is due to these crypts and villi. So the whole structure of the surface of the small intestine is a lot of folds, which results in a very large surface area. And then on these villi, there sits enterocytes that are responsible for taking in nutrients. And these cells have some structures called microvilli, which enlarge in the surface area even more. And then they have tight junctions, which bind the cells very tightly together. And these tight junctions secure that the only things that you get into the body are the nutrients that you're supposed to get into your body. And I will talk a bit more about these tight junctions later, as they are a crucial protein for this type of cell. And then if we look briefly just a bit closer to these tight junctions, then they are built up of a lot of different proteins, and one of them is called ZO1 or zoononcludensis 1, and another is called actin. And we'll touch upon these two proteins later on in the talk, as they are very useful for visualizing these tight junctions. And the actin is kind of the structural protein of the whole cell, and then it's also connected to these tight junctions. Yes. So the small intestinal wall or barrier is very tight, as I just mentioned, but it is still possible for nutrients and molecules to travel across, and this can be done in a number of ways. One is transcellular, where the drug will be able to just pass through if it has a specific structure. Another is a paracellular route, where it can go actually through those tight junctions. Some drugs are transported carrier mediated, where there are transport proteins carrying the molecules across. Then there can also happen transcytosis, where the cells take up the molecule and pass it through. However, this can also end in recycling of the molecule or even metabolism of the molecule. And transcellular, paracellular, and carrier-mediated transport are the three transport types that I've been working with during my PhD, and we will get back to these later on in the talk. So if we then look at the capillaries, which were the next part, when we went from the intestine, we went into the blood vessels, and they can be, depending on where in the body you're looking, then the capillaries or blood vessels have, the blood vessels probably have the same structure, but the capillaries can have different structures, different places in the bodies. And they can be continuous if you're looking at, so sort of the larger blood vessels, then they will be continuous so that there are no leakage of nutrients and cells and such. However, if you then are in the stomach, then the capillaries, they will be fenestrated so that the nutrients that you absorb through your small intestinal barrier then can pass into the bloodstream and travel further on in the body. And if you then look at the capillaries that are in the liver, then they are what's called discontinuous. So they have even larger holes, because in the liver functionally needs to clean the blood. So you will have you'll have a higher need for leaking out cells and toxins and such, so that these can be metabolized in the liver. And therefore, these kind of capillaries are discontinuous. And then for the model that I was trying to build my PhD, then I was going for like some capillaries that were either fenestrated or discontinuous, as I was working with the small intestine and the liver. Yes, and then the next structure of these three is the liver. And as I touched upon just before, the liver is this metabolizing tissue that can metabolize any toxins or drugs. And therefore it's structurally built so that there are different zones of oxygenation, because different metabolic enzymes have or are functional in different degrees of oxygenation. So this this change in the oxygenation along the liver tissue ensures that you have a lot of different metabolic enzymes active. So that is also something that we had to consider while building the liver part of the cell-based tissue models that I work with doing my PhD. So if we look at what is on the market or was on the market of other solutions before my PhD, so we'll say, you can say sort of the competition that I had, or the other I want to say other people working on it that I could draw some inspiration from. Then there were different approaches, many based on microfluidics, and one is this type where they have like a whole plate, so they have a lot of they can do a lot of repetitions at the same time, and then still have a tube of intestinal cells, and then look at what goes across to the other side. So the benefit that this model has is that you can do a lot of repetitions. Then there are other models, such as this one, where you see the 1, 2, 3, 4, where there are these multiple types of tissues, and that these tissues are coupled. So this is also something that we wanted to do in make a model of the small intestine and the blood vessels and the liver, and then have them coupled so that we can see how they interfere with each other. And then another approach with the microfluidics is this this one got on a chip, where you have these vacuum chambers that can stretch in the cell layer, so you get some mechanical actuation. So this is kind of what I tried to implement in my PhD project, from inspiration from what I had seen others do, to see if I could make something where I both had the possibility of doing multiple intestines parallel and having multiple tissues connected, and some mechanical actuation at the same time. Yes, so the hypothesis and aim for my PhD project were that you would be able to make a very good prediction of how much drug that you could absorb and have active in the body by connecting a cell-based model of the small intestine, the blood vessels, and the liver. And then, so that's kind of the hypothesis of the whole project. And then the aims were that it should be easy to do a lot of duplications, and that these 3 tissues should be coupled in the same system, and that there should should be able to incorporate some mechanical actuation. And then another thing I saw when I looked at what others had done is that then often the cells would grow on a plastic surface or a filter, which was hard. So I also wanted something that was soft to better simulate the tissues of the body. And then it should also be easy to use. So you can say that we had a lot of aims and were quite ambitious with what we wanted the whole setup to be able to do. But this was what I started and tried to do. So if we then go back to the outline, then I will start to touch upon the stiffness, how this was achieved. And I'll just mention it briefly and not go into the study that I have done on how to measure the stiffness and optimize the stiffness, but just briefly mention how or what we ended up with doing. So normally when you look at batch absorption methods, you would have these types of filter inserts where you have the small filter and then your cells grow on top. You can then add your drug molecules and look at how much of your drug goes through by sampling below the cells. And we drew inspiration from this, but then instead of having a filter, we replaced the filter with a soft hydrogel. So that this hydrogel was closer to the stiffnesses you would find in the actual tissues than this filter. And then the whole thing was done with 3D printed title plate inserts, which we designed ourselves. And you see the small hole here, which is where we then casted the hydrogel and then seated the cells on top. So we had the setup look like this. So very similar to what you would normally see with the cells sitting on top of a filter, but instead they are on top of a hydrogel. And then we could, as you'd normally do, add the drugs on top and sample below to see how much of the drug was able to be absorbed through the small intestinal cells and through the blood vessels and such. Yes, so that was just briefly the stiffness. Then I'll move on to biocompatibility. And in terms of biocompatibility, then in the beginning I had issues because I 3D printed everything myself, and the 3D print was slightly toxic to the cells, as you will see. So if we go through like a growing process, then at day 0 I would seed the cells, and at day 4 they would clump a bit and maybe start to divide. But then, as you see, they never really, even after 17 days, this type of cell did not really start to spread around. And by day 21, I started to see dead cells falling off, and then laying in here in the middle. Yeah, just to explain a bit more. So this is the 3D print, and then these are the gel parts. And these 3D structures that we have here are made to support the soft gel. That's why it's not just like a window. So this was one of the first issues that I had doing my PhD, and actually in general had for most of my PhD, as this bioincompatibility was a very large issue for me to solve during my PhD. But in the end, I figured out to do some vigorous steps of washing the 3D print, where it would lay first in some solvents to get rid of whatever residue that there was, and then lay in water to get rid of whatever was water soluble before addition of the cells. And then once I figured this out, then you can see here how the cells are dividing more, and then after 16 days they're then filling out the whole gel window so that it becomes an actual barrier. Yes. So that was the biocompatibility part that I had to solve. So now we have the correct stiffness, and we have 3D prints that are biocompatible. Then the next issue or task was to then build these 3 models. So the small intestine, the blood vessel, and the liver. And I'll start by talking about the small intestines. And I used these cells called CACO2 cells. And they grew on the hydrogel into a tight layer. And if we then see here, I have a graph of the growth of the cells over 3 weeks, and I have this tier, which is the tier measurements that are made, which are measurements of the resistance of the cell layer. So the higher the resistance, the tighter the cells are packed together, and this rise in resistance comes from the formation of these tight junctions. And we see this resistance is increasing over 21 days. And if we compare these CACO2 cells on a filter to what we have here on the gel, then the filter is much tighter. But in fact, if we then compare this with what you see in the small intestine in the human body, then they are actually both above the resistance. So you could say that maybe we should actually be using the cells down here, but at this point, the cells are not covering the gel or the filter at all. They are first covering at this point. And this is a general thing that you will often see with cells that you have in culture, that they are acting a lot differently than cells in the human body. But we're kind of achieving something which also develops the tight junctions and covers the filter or the gel, but does it in a manner where it is still what you could call too tight, but less too tight than what you would normally have. And if we then we stained for both for the actin to visualize how the cockatoo cells were in the hydrogel, and we can see that they're sitting close to each other with a large nucleus, and we also see a high expression of the tight junction protein between the individual cells. So now I will move on to the blood vessels. And for the blood vessels, we used primary endothelial cells, and they also grew on the hydrogel. You can see here they are lying like elongated cells. So compared to the enterocytes, which are just small round cells, these endothelial cells are longer, are more elongated cell type. And these we also stained for actin and for the nucleus to see the structure better. And you can see that they are long elongated cells, and you can even see the individual actin fibers. But you can also see that there are some holes in between some of the cells. So we are kind of achieving something similar to the fenestrated discontinuous or the discontinuous blood vessels with gaps in between the blood cells. But this tissue is also not supposed to be as tight as the small intestine, but is still supposed, or it's still interesting for us to investigate to see if any see if any drugs interact with the endothelial cells, as that will have an impact on the drug distribution. And from there, I will move on to the liver. And what is interesting to note about the liver is that it is structurally different from the two other tissues that we just talked about, where the small intestine and the blood vessels are like hollow organs or shaped like tubes. Then the liver is a firm clump of an organ. So we try to make a different kind of tissue, where we seeded the cells on top of the hydrogel. For the liver, we seeded them inside of the hydrogel to have this firm type of organ. And then we seeded them into this type of cylinder in the 3D printed insert, and then they had access to liquid and nutrients on the top and on the bottom. And then what I learned or could see was that I could grow the liver cells to these large spheres in the top and also in the bottom. But actually in the middle, we had like a depletion of nutrients. And probably what we also see is that we get this gradient of the oxygenation. So we would expect to have different enzyme function along this cylinder of liver, as we have a higher oxygenation level in the bottom and in the top and a very low oxygenation level in the middle. Yes. So now, just to summarize a bit before I move on, we have these three tissues built up. We have the intestinal layer of CACO2 cells and the endothelial layer or blood vessel layer with Huvec cells and a hepatic layer of HepG2 liver cells. And then the next task was to couple these three so that we can see how so we can see what it does to a drug in terms of absorption into the body to have these three connected. So for connecting them, I utilized these 3D printed plates that I had designed, and they could stack on top of each other. So like this, where you have the small intestinal tissue model on top, and then the blood vessel, and then the liver. And this could all then fit onto a tighter plate. Yes, this could all fit onto a tighter plate. And then we were able to then add a drug and then measure in the individual level how far this drug had traveled and what was, then you should all be able to measure in the sample what was the state of the drug. So you maybe were able to see that the drug could pass through the small intestine, but then see that it would be metabolized in the liver. So I used some different models or model drugs to test this out. And one was this molecule called Lucifer Yellow, which is able to pass through using the paracellular route. And then we used this to compare the performance of our CACO2 cells on the gel compared to the CACO2 cells on the filter. And what we then see, if we look at the permeability of this molecule, is that when we have cells on the gel and when we have cells on the filter, we have a very low permeability, whereas when we have just the filter or just the gel, then we have a high permeability. So we used this to determine that we indeed had, or we indeed could create a barrier using our model with the small intestinal cells on the gel. Then I tried to see what was the transport rate for the blood vessel, and I saw similar permeabilities for cells on a gel and for an empty gel. So this confirmed what we saw previously, that we have these gaps between some of the endothelial cells, so that it is this type of discontinuous capillary. And then for the liver, what I saw there was that there was a very low permeability. So this is probably caused by that you see that this is quite thin and this is quite thick. So maybe I had designed this part of my project, so the liver model, maybe too thick to actually be used at this point, as nothing would really pass through it. So that is you can say that is an optimization area, which I did not have time to investigate. And then I tested the model using furosemide, which travels through the transcellular route. And then what I saw there was that, again, it confirmed that the cells acted as a barrier, but were also able to, so this again confirmed that the small intestinal cells on the gel acted as a barrier, as they should. And then we looked into some other drugs. We tried with a product called valacyclovir to see if we could transport this across, because this has a very low permeability. So we were interested in protecting it before it was transported, to see if some of my colleagues, they worked on ways of protecting this product. But what we then saw was that all the product was converted into drug, which is not able to use these carrier. So we could conclude that our model of the small intestine worked as intended, that it created a barrier, but our ideas of protecting valacyclovir, so that it could be used as an oral, a better oral drug, were not really successful. Yes, so that was what happened. That part of the molecule was cut off too fast so that it could not be transported. But at least this showed that the model functioned as a barrier. Yes, and then I'll just touch briefly upon mechanical actuation before giving a conclusion. So for mechanical actuation, I worked on adding these whisks into the cells, or like, so this should then click on top and then whisk on top of the cell models to add some mechanical stress. And what we saw was that if we just have the normal cells, then they are looking like this. These are the endothelial cells. But if we then turned on these whisks and made a lot of turbulence in while they were growing, we could get this these even more elongated cells. And then we tried also with some different factors to add in to see if that made any difference. And you can see if we just add TNF alpha as a factor, then the cells are reacting slightly. But if we have TNF alpha and shear, then we get these very large elongated cells. So you could say that this is kind of the next step, or this would be the next step if I had continued working at D2U on this project, would probably be to implement this very nice endothelial cell model that we created to add this into this whole triple layer. But this was developed in parallel with me testing all those different types of drugs. Yes, so now we have kind of reached or achieved these 5 parts. And I will move on to a conclusion to kind of conclude on what I managed to do during my PhD. What the project ended up with having was these 3 different tissues that could be cultured individually, and then once they were fully grown, it could be assembled and coupled to this system, and then to test different types of drugs. And then it was relatively easy to use, as you can pipette in each layer of this model, and also achieved to have the cells sit on a soft membrane compared to a filter or a plastic. Then this project also created something which is scalable, as all the components used, all the components used, apart from incubators and such, were 3D printed and can therefore easily be adjusted to what needs you'd have. And as this is this whole system just can go on top of a normal titer plate system, then it is suitable for adapting to automation. If we look at some future perspective, so you can say what I did not have time to do would be to actually confirm this statement that I made with the gradient of oxygenation and the different liver enzymes, to actually test if my hypothesis with having different enzymes active is true. And then this whole model, say, for it to be a useful model, you would need to correlate what you see with what you see in vivo in a person or in a laboratory animal. So it would be nice to test a lot of drugs and then do a correlation with what is known about these drugs. And then another thing that would also be interesting to investigate is how exactly these 3 tissues, they interfere with each other, and how that has an effect on oral drug delivery. And then I would also need to look into, or the PhD after me would benefit from looking into some adherence molecules. That's the cells do not always stick that nicely to hydrogels. And then there's this whole time scale issue, as I talked about the liver part being very thick. So that's indicative of that it can take a lot of time for the molecules to diffuse through this gel compared to a filter. And then to actually implement this mechanical actuation that I started on a bit. Yes, and then I will just thank my former groups. And this is what the groups looked like when I was working at DTU. And I cannot guarantee if you that if you visit the Edron group or the FAST group at DTU that these are the people who are working there now. But at least this was how it was when I was working there. And that rounds off my talk. Thanks a lot for listening.
Thank you, Morten, for this great presentation. I think